So for this audio lecture, we'll be looking at chapter 4, which covers antibody structure and B cell diversity. So looking at the B cell receptor. So some of the basic properties of antibodies or B cell receptors are that they have 2 light chains and 2 heavy chains. They're also going to have a constant and a variable region, and we'll look at that in some slides. The variable region is where the antibody is going to bind, and so for each B cell, the antibodies and B cell receptors they produce unique antigen binding sites. So again, it lets them bind to different antigens, and more specifically different epitopes. They also have a hinge region, and that hinge region is flexible, so it allows for those variable regions to actually move a bit so that it can attach to either epitopes that are close together or epitopes that are further away. And again, we'll look at an image that demonstrates that. There are different classes and isotopes of antibodies. So there's like IgG, IgM, IgA, IgD, IgEs. And then with IgGs, there's actually isotype subtypes of those. And so for those different classes and isotypes, it's the constant region that changes. So we'll look at how the B cell can control which constant region is being expressed. And mRNA splicing is the key. So the B cell will have all the constant regions for the different classes, but then it will, when it's making the mRNA, it will splice in the constant region for the protein for the antibody to be produced. And then also antibodies have domains, and so when we're looking how they're folding, they're going to create these domains, and those domains have certain characteristics, and those domains can undergo editing, especially variable regions, especially the domains that make up the variable region. So here we have our B cell. Our B cell has membrane-bound antibodies, and you can see how those antibodies act as receptors, or the B cell receptors, and can bind to pathogens, foreign material, and again, they're able to interact with what we would call the antigen. Again, keep in mind, antigen would be the whole entire molecule. The B cell receptor, that antigen binding receptor, is actually only binding to a very specific portion of that. So really, it's these epitope binding sites that are interacting with the specific epitopes that they recognize. And B cells can undergo maturation, they can mature, and they can actually produce secreted forms of these antibodies. So instead of having membrane-bound antibodies, they can produce secreted antibodies. And again, we'll look at that a little bit later. And also additionally, when we're looking at activation of B cells later on in the semester. So here's our basic structure of an antibody, or an immunoglobulin, or IgG, more specifically. And again, you can see how in the green we have the heavy chains, and each of the heavy chains are identical to the other. So there's 2 heavy chains, but they're the same genes, same protein that's being made. And then we have the light chain, and the light chain's shown in here in the yellow. And so again, 2 light chains, it's the same chain for each of those yellow portions. You'll notice it's held together by disulfur bonds. There's also sites where carbohydrates can be added for modifications. And then you'll also notice that in the lower panel, instead of showing you the light chains and heavy chains, it's showing you the variable regions versus constant regions. So you'll notice the bottom portion is where the constant region is. And so this is constant, conserved, the same for pretty much every single antibody. There's going to be slight variations, but again, it's a little bit conserved in the different classes and isotypes of antibodies. What is really unique to each individual B cell is that variable region, because that variable region codes for the antigen, or really, again, what I was saying, the epitope binding site. So this is where the molecule, the portion of the molecule which the B cell recognizes, is going to bind. So in this figure, you can see how there's 4 polypeptide chains that make it up. So if we were to denature this protein, the immunoglobulin, the antibody, we would actually get 2 bands because they're identical, they're the same molecular weight, but there's actually would be 4 separate proteins. since they're proteins, they're also susceptible to proteases. So if we were to take a protease, that's actually going to cleave where the hinge region is, and that would actually create 3 fragments. So the interactions between the light chain and the heavy chain would stay, and then also the interactions between the constant regions of the lower constant regions of the heavy chain would also stay intact, but those hinge regions would be cleaved, cut by the protease, and so again we get 3 fragments. So we'd have 2 Fab fragments and 1 FC fragment. So just some fun facts if you're trying to denature some antibodies. Here you can see how we'll have multiple B cells, right, in our host, and so each of those B cells are going to produce different antibodies that can recognize different epitopes on a bacteria. So here we have 3 different antibodies recognizing 3 different epitopes. You can also notice how the space between those Fab fragments vary depending upon where those antigens are present. And also it would depend upon what type, class, isotype of antibody you have, how much flexibility those hinge regions have. But you can see how the one that's recognizing the rectangles on the kind of bottom right, those are spaced out further because, again, the flexibility of that hinge region. This is just showing you the basic structure of the different classes of antibodies. We will look at the different classes of the antibodies at the end of this audio lecture. But just to show you that, yes, they are different. So they have unique sites where you can have carbohydrates. So the green like hexagons are showing where you can have carbohydrates added for modification. You also notice that the number of domains that are making it up are also different. So you can see IgEs have additional domains that are lacking in other classes of antibodies. So if we were to look at the antibody as a ribbon model instead of that kind of blocky image from that we've been seeing so far, you can see that we have the different domains that are kind of coded to agree with what's in panel B. And you can see that we have 4 domains in the Fab region, and then we have also 4 domains that are down in the FC domains. So there ends up being 12 different domains that are interacting with each other in order to have the functional immunoglobulin. So as I mentioned, antigen binding sites, they recognize a specific epitope. So remember, antigen is the whole entire molecule. So if we're talking about a protein, an antigen can be, hundreds of amino acids long, but the epitope is generally between 10 to 13 amino acids long. And we'll talk about epitopes aren't always linear, so it's not always a continuous sequence of those 10 to 13 amino acids. Other things can act as antigens, such as carbohydrates, nucleic acids. So again, the specific molecules, atoms have to be present in order for it to bind to the antigen binding site. So an antigen can have multiple epitopes on it, so there can be multiple B cells and T cells that recognize different places on different locations on that antigen. So when we're looking at the antigen binding site on the antibody, remember that is made up also of amino acid sequences, right? It's a protein, and each amino acid has a different side chain. So if we change an amino acid that corresponds to the sequence that makes up the antigen binding site, it could change the side chain. So it can change what is able to be recognized, what the epitope is. And as I was alluding to, epitopes can be linear or discontinuous. Linear is where those 10 to 13 amino acids are right in sequence. Where discontinuous is there's an amino acid over here, there's an amino acid over there, there's an amino acid over there. But because the way the protein folds, it ends up interacting with that antigen binding site. And we'll look at that in just a moment. So here we can see how there's this term called multivalent, meaning multiple binding, multiple epitopes. And so we can see here on the left side how this antigen has different epitopes which different antibodies recognize. On the right, we have the same epitope, but it's repeated, so the antibody is able to bind multiple places. So the more recognition you have, the more engagement with antibodies, either as individual antibodies or different antibodies, is going to give you better recognition of the antigen, and then clearing out pathogen. So as mentioned, the epitope has to be able to bind into the antigen binding site. And depending upon the amino acids that make up the antigen binding site and the side chains, it can look different, right? So we can have a range from something that's a pocket that the epitope actually has to fit into. It could be a groove where there's only a certain portion that's actually directly interacting with. Or we can have, it doesn't always have to be like an indentation, it can actually be an extension coming out. So you see that on the right panel there, how that the side chains that make up the antigen binding site actually protrude out. So again, that the epitope it's binding to has to be able to engage those side chains. With the idea of linear and discontinuous epitopes, this figure shows you nicely how a linear epitope, again, it's a run of amino acids. I wish the image was a little bit more accurate and actually showed like 10 to 13 amino acids engaged with the antigen binding site, but hey, I'll take it. On the discontinuous epitope, you can see how there is amino acids that are not recognized by the antigen binding site. So again, it's not a continuous recognition. It's a portion here, and it could be 3 to 4 amino acids in one section, and then another 3 or 4 amino acids in another section, and then 2 more somewhere else. And so that means that with discontinuous epitopes, if you denature the protein, you lose that recognition. With linear epitopes, you can actually create peptide libraries, and peptide libraries are where you take the sequence of a protein and create peptides from it, and you do 10 to 13 amino acids long, and you make sure you have complete coverage of the antigen, and then you can feed those to B cells or T cells to see whether they become activated. What peptide do they actually recognize? So again, a linear, you can use that approach. For discontinuous, that's not going to work because you're actually looking for a portion here, a portion there, and folding is really important. We'll look at a technique called virus scan. Virus scan kind of helps with discontinuous epitopes as long as the portion of the protein that's being expressed is long enough and needs to be folded to create that discontinuous epitope. So again, discontinuous epitopes are a little more difficult to study, just because peptide libraries are kind of a go-to tool to identify epitopes. So we can actually, produce antibodies, and they can be used for different techniques and even treatments. So given the knowledge we have on how B cells are able to produce antibodies, we can create monoclonal antibodies by producing what's known as hybridomas. And we'll talk about what hybridomas look like. These isolated antibodies can be used for different laboratory techniques, such as cell sorting, ELISAs, Westerns, etc. Ones that we will look at in Journal Club, and you have done yourself in other classes if you've had a lab component. And they can also be used for treatment for various illnesses. And we've actually seen that in the news for monoclonal antibody therapies for COVID-19. So what happens in producing a monoclonal antibody, what you would do is develop B cells that recognize the antigen you're interested in. So whatever the protein you're interested in, generally what you do is inject mice with that protein. They end up developing antibodies to it. So you screen for antibody production, and then you isolate B cells from the mouse. And those B cells that are isolated usually go through another screening process to make sure they are producing antibodies of interest. And those B cells get fused with myeloma cells. Myeloma cells are immortal. And so when you fuse them together, that actually makes the B cell immortal too. The cell, that fused hybridoma, has the genes from the B cell, so it has the genes to create the antibody. A lot of times there's a drug induction, so you can turn the expression on of the antibodies. So then you can purify the antibodies. And again, hybridomas and antibodies would be screened and selected for to make sure that they are expressing antibodies for the protein of interest. But because they're immortal, they become easy to culture. So you would have a source of those antibodies. So a lot of those, companies like Abcam and such that produce antibodies would actually have a library of hybridomas that they are producing antibodies from, which then you as a consumer can purchase those antibodies to use for different techniques, such as flow cytometry, also known as cell sorting or FACS. So we can use these antibodies to label the surface markers on the cells, and then using an apparatus with a laser, they can detect different dyes that have been tagged to the antibody. So again, antibodies could then be modified to be used in a research type setting. You're probably familiar with Western blots. So kind of the end part of a Western blot where you've taken a lysate and run that out on a gel and transferred it to a membrane. You're using antibodies that specifically recognize the protein of interest. They can be used in microscopy. So again, you can take a section or cells that are in culture and look at them under the microscope after you've added your antibodies and detected the antibodies to see where your protein of interest is found. And they can also be used in ELISA to look at whether cytokines are being produced or different chemokines are being produced. As mentioned, monoclonal antibodies are also used for treatments nowadays. So we'll do a class activity related to monoclonal antibodies. But there are a number of different forms. So some are directly produced in mice. So they produce antibodies to like human proteins, and those antibodies then can be used for various treatments. A number of monoclonal antibodies are for cancer, but there's also monoclonal antibodies for infectious diseases, also things like allergies, asthma. There are a number of antibodies looking at that. But the original antibodies were derived from mice, and since then there's been some tweaking of the antibodies so that they are being, you can take these hybridoma cells and they can actually produce human antibodies. And again, we'll talk more about monoclonal antibody therapies with the class activity that we'll do. So when we look at B cells, they really have an endless repertoire of antibodies that they can produce. And so the question kind of becomes, well, how do they do this, right? So if all these B cells originating from the hematopoietic stem cell have the same genome, how are they actually able to produce all these different antibodies that recognize different epitopes, different antigens? And the way they do that is with their gene segments. So with B cells, they have 3 genes that code for antibodies. They have 2 light chains and 1 heavy chain. So the 2 top genes here are light chains, and then the bottom one is a heavy chain. And what you'll notice is that there's these different segments. So there's the variable segments, which are shown in red. There's the diversity segments, which are only found on heavy chains, which are shown in green. And then joining segments, which are shown in yellow. And C, or constant segments, which are shown in blue. And so what's happening during B cell development, which we'll talk about later on in the semester, they will use a light chain and the heavy chain. So they have 2 light chains to pick from, right? So they could pick the first one, which is kappa, or they could pick, or that's lambda, or they could pick kappa. And then they are selecting which variable segment they're going to use in a heavy chain, what D diversity segment they're going to use, which joining segment they're going to use, and which constant segment they're going to use, right? So, so you can see here, like if we just focus on lambda, which is the top gene, for that light chain that's going to be used to make an antibody, we could pick variable 1, joining 2, and that comes with constant 2. Or we could pick variable 2, joining 1, constant 1. When we look at the heavy chain, we could pick variable 3, diversity 20, joining 2, and then it keeps all the constants. But depending upon what the expression's needed, it would pick a constant to actually express. So this starts to add to the diversity that's possible, and that's what we're going to kind of look at for our next kind of slides. We'll go through some background information about the different genes, but then we'll look at how these different segments get selected. And again, some of this will actually carry over into our next unit when we're looking at development. So just giving you some background information about the different genes. Remember that each antibody is going to have 2 identical heavy chains. We only have 1 heavy chain gene, so we know that is the gene that's going to be selected for. And then remember that there's going to be 2 identical light chains. So this means it's either going to be kappa or it's going to be lambda, but it's not going to use both, okay, for the final antibody that's being produced. When we look at the number of segments that are present, you can see that light chains have a high number of variable regions, no diversity region, around 5 joining regions, and then their constant regions for kappa only has 1, but for lambda has between 4 and 5. For the heavy chains, again, the most segments are in the variable region, but they also do have a number of segments, 23 segments in the diversity. Joining has 6, and then constant has 9 because there are 9 different classes or isotopes that the B cell can produce. So if you did some math and did all the calculations, again, you'd find out that there's really an endless number of combinations that you can have when we're looking at the final antibody that's actually being produced. So again, just a little rundown. Kappa, again, it's going to have variable joining and constant region. For lambda, it also has variable joining and constant, which one way to determine which gene is which is lambda actually has the joining and constant paired. So if you pick joining 1, you get constant 1. So for kappa, those are independently selected. So you can get joining 1 and constant. It actually only has 1 constant. So usually what happens is that kappa is selected. So kappa during development, again, we'll talk about this later on in the semester, kappa is the first light chain that gets selected to be expressed. And if kappa doesn't work out, then you use lambda. So lambda is more of a default light chain. It's a backup plan. So you can see in mice, if we look at antibodies, there's going to be less than 5% of them actually use kappa. In humans, it's a little bit higher, where 40% of light chains are lambda, which means 60% of light chains are kappa. So again, kappa is the default, like you go to first. Lambda is the backup plan. And then with heavy chain, they have all 4 segments. So variable, diversity, joining, and constant. And again, remember that the constant segments codes for the different isotypes, different classes of antibodies that are produced. So again, how this kind of works out to be is that with the light chain, you have your variable, joining, and constant. And so again, what's going to happen is that they're going to select for a variable and a joining, and that actually gets rearranged and joined together. So you can see here down at the bottom for the light chain how that space between the variable and joining has been removed. So that's going to end up, coding for your functional light chain. When we're looking at the heavy chain, what's going to happen is the D and J is going to join together. So you can see how that green portion and the yellow portion, that space between it, has been removed. And then there's this kind of second round where then we have a variable selected. And so you can notice that the space between the red and that green disappears on that second round. So now we have a VDJ that's been selected and rearranged, so it's joined together. You'll notice that you're keeping those constants on the heavy chain. So it's not that it's selecting for each one. But again, what you'll notice is that what is in the germline DNA is different than what is in the final B cell after it's undergone gene segment rearrangement. So this in lies, the misconception of all our cells have the same genome. No, the germline DNA might be the same, but you as a human being who's lived on this planet, if you've had B cells undergone gene segment rearrangement, which you have, then your B cells have different DNA from each one. So each of your B cells, unless they're a clone of a B cell, have different DNA in them. And your B cells have different DNA in them than T cells, because T cells are going through a similar process also, which we'll look at. And so each B cell, each T cell has a different genome. And then think of all the other cells in your body. They have shared genomes. So this process of gene segment rearrangement is unique to these lymphocytes, right? So again, a little bit of a misconception that all our cells have the same genome, because we have T cells and B cells, and they're each going to have different genomes. So the mechanism, the details of how gene segment rearrangement or VDJ rearrangement happens is it's directed through these signal sequences. So there are sequences or recombination signal sequences, RSSs, that are found in the sequence of your light or your heavy chain genes. And that flanks the antibody gene segment. So that variable segment that's going to be selected for is going to have these recombination signal sequences that allow for the enzyme to be able to recognize this is where I need to bind to, so I can hold the DNA in position so it can be cut. So these recombination signal sequences are known as nomers or hapmers, depending on how many are in the sequence. And we're actually going to look at a figure that shows this. So here we can see we've got this variable. This is one of the variable segments, like in the lambda on the top, and one of the joining, okay? And you'll notice that there's these different segments in there. There's the 7, which is our hapmer, and then our N base pairs, which is shown in the purple, that's our nonomer. Okay, so again, remember, because you're going to see a figure in a couple seconds, or a couple minutes, that's going to show you these again, but with an enzyme bound to it. So 7 and 9 that are close to the variable are one kind of binding anchoring sites for the enzyme, and then the 9 and the 7 close to the joining is another binding site. So it means that information in between, which is in the gray, is kind of, it's not necessary. Now, that gray portion changes, right? So this is just showing you the variable and the joining kind of somewhat close together. But remember, if we're picking variable 1, there's all the other variable segments there, right? And so that gray area is where those other variable segments would be contained. So again, you can see this pattern is shown for both light chains and also the heavy chain. So the enzymes that play a role are RAG1 and RAG2 recombinase. So RAG stands for recombination activation gene. So this codes for RAG, the enzyme, which is a recombinase. And so again, it's going to bind to these recombination signal sequences and hold the DNA in portion. So RAG1 specifically does that, so that RAG2 can come in and cut the DNA so that it can then be fused together. So here we see in our image how we have RAG1, which is shown in the green, and it is binding to the nonomer, and that is the 9, and then the hapmer, which is the 7. So again, think back to that figure we just saw. So there's those anchoring points, the 7 and the 9. And again, RAG1's binding to that, and on the variable side and also on the joining side, holding that together, RAG2 is also in there, and RAG2 is actually going to cleave right there where that between the hapmer and the gene segment that's actually going to be utilized. So that means everything on that loop gets cut out. It's gone. So before this moment and after this moment, the genetic information of this B cell has been altered, is different, right? So again, that's like the biggest thing. If you can have that appreciation, I think I've made a difference. So that signal joint there, or what they're calling the signal joint, again, is going to be removed, and depending upon what portions are being utilized, which variable regions, which joining regions are being in the coding joint, it changes how much DNA is being removed, what gene segments are being removed, okay? So again, this is just showing you, okay, we're not going to use variable 2 and 3, so that's being removed, right, from it. I included this figure just because this is from a different textbook, but shows a little bit more three-dimensional, the interactions between RAG1 and RAG2 with the DNA. So again, you can kind of see that what they're calling the interweaving DNA, that is going to be that what is being excluded is being removed. This is so this is where some students start to like glaze over. So this is getting like too much. Just think big picture. These are like added details. I put in bold some of the terms that you should know because yeah, that kind of stuff is important. But what happens is we end up having, we're going to have that cleaving, so we're going to lose the hapmer and anything that's in there. So that's what the RAG complex is going to be doing. What then happens is that we get these hairpins that are formed. So you can see these little loops at the end. So then what happens is those hairpins open so that you end up having a single strand of sequence on the D and on the J, or the V and the J, depending upon whether you're looking at the heavy or the light chain. Okay, so this is showing heavy chain because D is standing for diversity. And so again, what's going to happen is we have that TC hairpin GA, but that's going to flip open. So then we have TCGA, and the same thing's happening on that joining side. Then what happens, so this ends up being known as P nucleotides, okay? So they actually create a palindrome, so that's where the P comes from, palindrome. Then we have TDT, which is a transferase enzyme that comes in and randomly adds nucleotides. So this stuff is not coded. So again, now the cell has a second change to its genetic information because we're adding these N nucleotides. These additional nucleotides are being added, and that is so that we can line those up. So what happens is these strands end up pairing where these base pairs will actually meet. So these are just randomly added to the ends of our P nucleotides. So we've got these N nucleotides have been added in randomly, and so by chance they end up pairing together because base pairs pair. Right? Remember intro bio. And then any of the ones that don't match get removed by exclunucleases. And then we have ligase coming in, DNA polymerase coming in to actually ligate and seal, if you will, this fragment. Okay, this coding joint. So lots of different enzymes involved with this, and a little bit different than when you're like had to memorize DNA synthesis, right? And different than when you're looking at exonucleases, restriction enzymes in their function. So again, just keep in mind. But everything we've talked to this point really leads to having a wide repertoire of antibody diversity in these B cells, okay? So, and then keep in mind at this point, these B cells have not seen antigen, so they're naive. They are just randomly kind of picking what segments to pick. So just because of the number of combinations of gene segments, that can give you different possibilities. And then combinational diversity, because we can pick different light chains. So we have a heavy chain, but that heavy chain can pair with different light chains. And then adding, having these P nucleotidases added from the hairpin turn, those are going to vary from each, depending upon what variable region, which joining region, which diversity region is being selected for. We also have the exonuclease trimming, so they can trim different nucleotides, right? So that's going to give us diversity in what ends up being in that coding joint. And then also with your N nucleotide addition, right? So those are just randomly being added, and so that adds diversity to that, to what the final genome is of that naive B cell. And again, it has not seen antigen, so we really don't even know whether this is a quote unquote good antibody, a good B cell receptor, because it may never see that antigen. It may never come across its epitope, and that would be a sad story, but don't worry, we'll talk about B cell development. So another process it can undergo is mRNA splicing, and this is where the constant region gets selected for. And so depending upon the cues that the signals that the B cell is getting at various points after it's been activated, it can select for a class or isotype of antibody that is best suited for what it's dealing with. And again, we'll talk a little bit more about that both during in this audio lecture, but also when we're talking about it later on with B cell development. And so again, it can splice in the constant region that it needs to produce. Okay. Now this process is known as class or isotype switching. So it's able to switch and change where the splicing happens, so a different constant region can be expressed. B cells are also able to co-express, so they're able to edit the constant region so that they can express both IgMs and IgDs at the same time. So one of the stages of B cells that we'll look at will actually look to see whether we're actually co-expressing or are we doing a single expression. And so you can see here on the left panel how we're going to use constant mu, and that codes for IgMs. Where if we're going to code for IgDs, we're going to pick constant delta, and so that's the dark purple, and so that's going to then code for IgDs. Just to kind of jump forward from the textbook to kind of wrap up the isotype switching, activation-induced cyclodinases, or AID, plays a role in isotype switching, and we'll also talk about somatic hypermutation in just a moment. So it's a critical enzyme that will actually target the specific constant region that needs to be expressed, allowing for switching of that region and also splicing out the portions that don't need. So very similar to what we saw with the recombinase, where you can see how we create this loop and then that is excised, removed, so that, but this is at the RNA level, so that then we're only producing the protein portion of the constant region we want to actually express. Now, to have a functional B cell receptor, we also need these two smaller proteins to be expressed, Ig alpha and Ig beta. They are co-receptors, so they actually will form disulfur bonds to each other. They're complex, and they also interact with the B cell receptor, the immunoglobulin. And so, again, really important that they're also present because they're going to be important for cell signaling events that we'll talk about later on with development. So with antibodies, they're able to either be membrane bound or secreted. And so this is also done by RNA processing. So you can see here how we have this orange portion here on the transmembrane when we're looking at the coding. And so when we are making the messenger RNA, if we splice out that portion, it actually will lead to the expression of a transmembrane portion. So this leads to a transmembrane IgM, which is going to be membrane bound. Where if we want a secreted IgM, if we include the secreted coding sequence, that is going to allow for one that doesn't have a transmembrane portion, so it would actually be secreted out. So again, depending upon which sequence is coded in the message RNA, that changes that bottom portion of the protein of the antibody, and then determines whether it has a transmembrane domain or whether it is not attached and is secreted. Another way that we can get variation in the antibodies is through somatic hypermutation. So this generally happens in the VJ region, which is where you're going to have interactions in the antigen binding site. So this figure just shows you how that VJ region has a high number of mutations that can occur. So again, that aid enzyme plays a role in having mutations occurring. Mutations means that we're changing the amino acid sequence. Changes to the amino acid sequence can change the side chain, and when we change the side chain in the antigen binding site, that changes what it can bind or how strong it can bind to something. So what happens over time, B cells actually start being selected for that. We end up selecting for B cells that make higher affinity antibodies, and it's through these somatic hypermutations that we end up having higher affinity antibodies. So we can see here after vaccination, if you look at where the red dots are, these indicate amino acids that have been changed from the original sequence. And then if you look at 2 weeks after primary immunization, you can see there's additional mutations that have happened against somatic hypermutations. So the B cell is always trying to edit and improve the antibodies. Sometimes they make an edit that's not higher affinity, it's actually lower affinity, and those ones just aren't going to go on and survive because they're not going to get signals, they're not going to get to interact with the antigen, because higher affinity antibodies are going to end up binding to the antigen, to the epitope. Okay, so there's always this editing that's going on and this improvement. And again, things like vaccinations and boosters, exposures, changes the genome of the B cell from what it was previously. The last portion of this audio lecture and these slides are looking at the different classes and isotypes of antibodies. So you should be aware that different classes and isotypes have different properties and different functions. So they play different roles in an immune response. So looking at the different classes and what's referred to as subclasses or isotypes, we can see that they have different molecular masses. The serum levels are different. So some of them are going to be found at higher concentrations in the serum versus others. So for example, something like IgDs, we don't tend to find those in high concentrations in the serum, but IgGs are usually secreted. They're produced by plasma cells, which have undergone B cells that have undergone maturation. So those are a lot of times found in circulation in the blood. Okay. They also have different half-lives. So you can see IgGs are pretty stable. So on average, about 20 days, if you exclude IgG3s, where other antibodies like IgEs and IgMs have a much shorter half-life in serum. And usually those ones are sometimes found in tissues. IgMs a lot of times are attached to B cells. So again, you wouldn't find them, one, at high concentrations in the serum, and their survival in the serum, if they are present, isn't going to be high. And then looking at the functions of the different isotypes of antibodies, you can see that there's some that are really good at neutralizing. So both IgGs and IgAs. IgA plays a role in mucosal immunity. They're good at neutralizing like toxins or viruses and bacteria. Opsonization, this is where you get the labeling with antibodies and leading to phagocytosis of the pathogen. So again, IgGs are well known for that function. IgGs are also good at being able to activate natural killer cells to induce killing. So you can see IgGs, there's a lot of great things they can do, and they're usually found in the second wave of an infection or a B cell response. So IgMs are usually the first ones. And so you can see that with IgMs, they actually lead to the activation of complement system, which makes sense. So if we're looking at okay, we haven't really seen this antigen before, so this is like a naive B cell producing IgMs. What can they do? Or IgGs even. What can they do? Well, they're playing roles with the innate immunity, so activation of complement system for IgMs. IgGs are activating basophils, so both of those are very innate processes. I just want to point out IgEs we'll talk about later on in this semester in Unit 6 if we talk about allergies. So they bind to mast cells, and that leads to mast cell activation. They can also bind to basophils. So both mast cells and basophils release histamine. So if we think about an allergic reaction, there's histamine release. So we take antihistamines to counter those effects. One of the properties of antibodies are some can actually cross into the placenta. So you can see some of the IgGs are able to cross the placentas, but other ones aren't. And also just epithelial layers. IgAs are good at crossing epithelial layers. Again, they're important for mucosal immunity, so being able to track down that pathogen is really important. And then you'll notice a number of them are able to diffuse into extravascular sites too. So I don't really expect you to memorize these different classes of antibodies, but you should just be aware that they can be found in monomer. They can be found as monomers, or they can complex together. So IgMs can actually complex together as a pentamer. IgAs are found as dimers. And then another important concept is I had mentioned about that hinge region. So you can see that that hinge region allows for different movement of the Fab regions of the antibody. So those arms can what's called wave, they can wag, they can bend, and they can rotate. So again, that can change how it's going to interact with the epitope, and that can also allow for multivalent binding, where you get binding of both Fab regions, both antigen binding sites on the Fab region. With the different subclasses, isotypes of IgG, that hinge region is different, so how that can flex and move also is different. And just for some completion, just some of the properties of the 4 subclasses of IgG, and their roles. And again, I would prefer you have the big picture pieces, in looking at the overall classes versus these different isotypes. So hopefully this was useful. If you do have any questions, please contact me, and we'll be going over some of these in class, and we have a class activity that we'll be doing this, and journal club that will reinforce all this information. So have a good day.